How many people are your lab?
8 PhD students, the lab manager, 2 master's students, and I've lost track of undergrads at this point. Like we're like something like 25.
I remember oscillates when you contact the surface. No, we didn't do it all on the saw.
Yeah, it's having a good opportunity to take a lot of good students. A lot of them are funded through fellowships or through my grants. So it's a machine at this point.
I was like, oh, do you know the name? And she was
And
like,
then some other stuff.
are there 2 of you? She said there are 2 of you.
Are you gonna talk about matrix remodeling
And I
today?
was like, I love her.
That's what she said. She said.
Calcification, I think, counts.
She said it prevents pollution calibration. And you should meet Steve Jones. He's here. He really likes Dave. I also know that's what I got here. And he was like, I've never met him. He seems cool on Twitter. I was like, he's looking at ventricular remodeling.
Cool.
So I thought it would have been a good guy, but he's now in med school. So I may have known him. Can you meet me from script?
There's pretty a lot of good cardiovascular research down there. Yeah.
He said, I believe it's not.
Yeah, it's pretty well known.
It's like a heart transplant. He said, I feel like so many clinics say that we're the first to use, but they did something. They were the ones to do something.
There's like photos, card
In South Africa and then Houston.
All right, everybody, let's go ahead and get started. Thank you for taking time to come in today. We have a visiting speaker for you. We got Dr. Josh Hutcheson from Florida International University. He's going to come here today and talk to you about his lab and his progress, where it's taken him in the context of molecular, mechanical, material mechanisms, and how he applies it to cardiovascular diseases. Today, just after the seminar, if you are interested, there will be people who stick around. And I'll let you know.
Thanks for having me. Happy to be here. I've known Dr. Chen for a long time, so really happy to be here and see all the cool things that y'all are doing. I'm gonna start out actually before I forget with a recruiting pitch. So I'm also our graduate program director in the department. So if any of you are undergrad students looking for grad opportunities, or if you're grad students looking for postdocs or looking for faculty, we're hiring at all levels. We're a big public university in the middle of Miami. This is actually the middle of our campus. It's a really nice place to live. The 4th largest university in the nation. Our department has 45 PhD students, 14 tenure track faculty, research expenditures last year about $7 million. So we're a big, really active research department. So if any of that's attractive to you, let me know and to talk to you more about what we've got going on as a department. But today I'm going to focus on what we're doing in our lab. And if you get bored by any of this, we have a big lab, as I was telling Joe. And if you get bored by any of this, just wait a couple minutes and I'll move on to something else because we kind of have academic ADD. We're a little bit all over the place, and I'm going to try to cover all of it. I have 8 PhD students, and my goal is to cover all of their research. So we'll see if I can do that, but also tie it together. So my sister designed our lab logo, and because I do cardiovascular research, I told her to make it look like a heart, an anatomical heart, so that I can use it in my presentations. And we focus on really 2 tissues within the heart. The aortic valve, which if you don't know is located between the left ventricle and the aorta. Make sure the blood flows in one direction out of the heart. These little leaflets in that valve have to open about 3 billion times in a lifetime, so very mechanically active environment. And when it gets diseased, it can't open as well, sometimes can't close as well. It puts a lot of stress on the heart and it can lead to heart failure. So we'll get there eventually. First, I want to talk to you about our work more in the coronary arteries, arterial diseases, things that lead to issues like heart attacks and strokes, things that lead to hypertension. And we spend a lot of time thinking about atherosclerosis, which is the main cause of heart attacks, when these plaques, these fatty plaques that you build up in your arteries rupture and you get this sudden thrombus that forms, and that cuts off blood to your heart, right? And that's when you have this major acute cardiac event. So if we look at the risk of that happening, the best predictor of having one of these major events is the formation of calcification in your arteries. Okay, so this is bone-like mineral that forms. So you can see On the left graph there, as the total calcium in the coronary artery goes up, the more likely you are to have a cardiovascular event, the worse your mortality is. And especially in folks with diseases like chronic kidney disease, this also holds in diabetes. So you get these other conditions. Calcification greatly predicts your survival. So what we're looking here is a survival curve, looking out, whatever that is, 7 years or so. If you have no calcification, you can see that most people survive well out that time. Whereas disease-matched controls, people with medial calcification in the medial layer of their arteries or the intimal layer, you can see how drastically your survivability drops. The predictive value of this was so great that in 2018, the American Heart Association kind of reversed course, where we were almost getting to the point where statins, lipid-lowering drugs, start putting those in water. I mean, it was really, they were prescribing it that much, right? They said even if you have moderately high cholesterol, if you have no detectable calcium in your coronary arteries, you can withhold statin treatment because your risk is so low of having an event. That was a major reversal. So this calcification also, we think, so it's really confusing what it does and why it's such a big deal. I mean, you can imagine if you have a lot of calcification in your arteries, that is going to put a lot of stress on your heart because it's trying to pump against these very hard vessels. And locally, we know that there's a mismatch in material properties. This local bone-like calcification, as opposed to this hyperelastic tissue that's around it, can lead to rupture of those plaques. But the problem is, once it's there, can we get it to go away? Right? You can imagine getting that bone-like material to go away is very difficult. So this is just showing you one of the major problems still in cardiovascular medicine is what we call residual risk. Okay, so I mentioned them before, these lipid-lowering drugs. They have greatly improved cardiovascular health. They are widely prescribed now. But this was a study from a few years ago. I think my reference here is animated for some reason. From about 10 years ago now, looking at the effects of statins, and you can see these are clinical trials across the board with thousands and thousands of patients, right? The yellow bars are the folks on statins, the blue bars are the folks who aren't. And you can see in every trial, statins lower your risk of having some kind of event. But look how high those bars still are in the yellow bar side, right? So even taking the statins is not taking your risk to zero, and sometimes only lowering the risk, by a fairly small margin, right? Similarly, lifestyle interventions like smoking, right? If you're a smoker, once you stop smoking, your risk remains elevated for at least 5 years beyond cessation of smoking. Right? If you're a heavy smoker, it doesn't return to baseline for about 20 years. So that's a big issue that we don't know how to solve yet. Once you have some kind of cardiovascular modeling going on, how do we reverse it? And we think it's this presence of calcification that's one of the keys that takes a long time to resorb that. So that calcification starts when these smooth muscle cells that are in your arteries, they're very plastic. They're very stem-like, and they can adopt very different phenotypic profiles. One being an osteochondrogenic profile. So the group at UCLA, led by Linda Niemer's group, has really studied this, but these cells become almost bone-like. They start expressing things like RUNX2 and alkaline phosphatase, these proteins that are well known in the bone field, and depositing this matrix. But if we think about atherosclerosis, we have these smooth muscle cells there, we have some fibroblasts there, with this huge inflammatory environment. It's really messy. And reversing that calcification is probably going to take more than just targeting these smooth muscle cells. You're probably going to have to have some kind of holistic treatment that both reduces inflammation and targets that calcification. So that's been one area of focus for us. One of the mechanisms that we started looking at is the relaxin hormone. And the reason for this is, if you're not familiar with it, relaxin is very heavily involved in remodeling in tissues during pregnancy and in postnatal recovery from pregnancy. And so I've always been fascinated by that you have these huge tissue changes that go back to something like they were before in adulthood, in an adult female, right? That's something you don't see very often. Adult tissues are usually very bad at themselves, about returning back to some baseline. And the idea is, if you can harness that power in other tissues, then perhaps you can then do the same thing, say, in the cardiovascular system, right? And so I have a collaborator at FIU who has focused on some of these relaxin mechanisms. This relaxin hormone is very anti-fibrotic, and it can both control inflammation, which I told you is a very important part of atherosclerosis, but also things like fibrosis, which is very closely related to calcification. However, previous iterations of this therapy, people have been trying this, have used antibodies for targeting these pathways or peptides for targeting these pathways, and those aren't very amenable to very chronic diseases, right? Where you might have to go in and get an injection every now and then, or then they can be very expensive. And so my collaborator worked closely with the NIH, NCATS, to develop a class of molecules that activate this relaxin receptor. So these are all small molecules, and we started working with him very early on in my time at FIU, and showed that ML290, one of their lead compounds, that we could both, this is in cell culture, I told you the calcification and inflammation, these two parts of the pathway that we're trying to we can inhibit calcification, which is what you're seeing on the left-hand side, but perhaps more importantly, we can cause macrophages, these inflammatory cells in the plaque, to become anti-inflammatories. They release reduced expression of like IL-1 beta, which that's one of the things that a lot of folks are trying to target in atherosclerosis to reduce inflammatory burden. So in vitro, we show that we can do this. So we do a lot of work with mouse models. We use this atherogenic mouse model. It should be noted that this small molecule only activates the human receptor, which is actually kind of nice. So we can do a lot of controls with that. But we made a mouse that expresses the human relaxin receptor. And so it won't activate the burden receptor. So it only works in that human receptor. This is a transgenic mouse. It's also deficient in something called apolipoprotein E. These mice can't process cholesterol. So they get hyperlipidemic. They become, we feed them a high-fat diet for 25 weeks. They become very fat, and they're happy, but they develop severe atherosclerosis as well right during that time. And we showed that with this ML290 molecule, small molecule, when we start giving it to them after the atherosclerosis is already developed, so it takes about 15 weeks for atherosclerosis to develop, we give them this small molecule, we can reduce calcification. So this is a dye that we inject at endpoint that shows us where the calcification is. So what you're seeing here is a whole mouse aorta. This is the aortic arch. This is where the carotid branches are, down into the femorals. And so where you see more signal, that's more calcification. And the ML290 treatment reduce that calcification. But like I said, what we really want to do in this study is understand, is that calcification reversible? The idea is those anti-inflammatory macrophages, they can become something that looks like osteoclasts. They can express the things that resorb bone mineral in your bone. And the way we started looking at this is that what we could do is we can inject different dyes that calcium mineral at different time points, right? So we have a blue dye that binds at one time point, a green dye that binds at a different time point, red, and then this osteosense, which is near infrared. And so it's kind of like looking at rings on a tree, right? As that tree grows, you can then look and see how that calcification goes. Because that you inject the dye, it only has a half-life of maybe a few hours. It binds to the mineral, it goes away. A few weeks later, you inject a different dye, and you can see that mineral growth. You can see that down here in the bottom where we have the green would have been the one. So there was no dye at the blue time point. This is too early for calcification. We see a little bit of calcification here in green. By the later weeks, you're looking at kind of a section here. It's out here, and then it's kind of grown this way out by 25 weeks. So we started giving the ML290 treatment at this green time point. So you would expect at least the green bars to be the same between these two groups, but they're not, right? And we actually see reduced red as well. So we think that we're having some kind of effect here where we're actually at these if we have early amounts of calcification, we might be able to resorb that, get rid of it, and also then prevent the progression of future calcifications. This is an area that's led by Anna Valentine, a PhD student in lab, like I said, it's collaboration with Dr. Agulnik in the College of Medicine. So we're really further exploring this and looking at associated mechanisms. So we're fairly excited about this because, again, this is kind of a bit of a holy grail in calcification in cardiovascular medicine in terms of how to get rid of this calcification. Now I want to focus in on just a bit on what causes that calcification to form in the first place. I told you that these cells become somewhat bone-like, right? They have these phenotypes of osteoblasts. If you look closely at this mineral, so this is work that we did during my postdoc when I was in Boston. This is actually from a human plaque. You look close at this mineral. So the mineral is in more orangey red here, and the more organic material is in green. You see it's not smooth. I mean, you might think of bone being very smooth, but it has this very fine substructure on the scale of millimeters. And it turns out that what this is, these are little extracellular vesicles. So some of you might know about exosomes, right? So cells release these vesicles. That's how they talk to each other. Well, it's been known since about the 60s that in bone, the way the mineral becomes nucleated to begin with is these vesicles are released and they serve as a little reservoir for calcium and phosphate to come together. So you have to get those ions close together to start the process. And so we've observed the same thing happens in the cardiovascular system when mineral is forming. So this is computational work that we do with Prim Shaffigan, who's a very talented molecular dynamics guy in physics at FIU, showing that these vesicles have very unique properties. They have a very negatively charged membrane that allows them to really bind that calcium, and which then can interact at the surface of the vesicle with phosphate and form this structure. Okay? But we're focused mostly on, well, how do these vesicles form in the first place? And is there a therapeutic target there that we could perhaps alter their formation to maybe, again, treat calcification? And so one thing that we know, again, from some mechanistic work that we started during postdoc back in 2016, was that the formation of these little vesicles, talking 100 nanometer size, we had to do the computational work, right? This is below traditional imaging resolution limits for an optical microscope, right? So 100 nanometer size, those vesicles rely on the presence of a specific protein in the membrane called caveolin-1. Caveolin-1 is a scaffolding protein in the membrane. So your plasma membrane of your cells have these little dips in it, in certain places, like here. These are called caveolae, which is Latin for little caves, right? And those little structures, they're very important. They aggregate a lot of receptors, but it also seems that these vesicles might form from those locations. If we knock down caveolin-1, we reduce calcification and activity of a certain enzyme called tissue non-specific alkaline phosphatase. It's an important enzyme in the calcification process. I'm going to bring that back up over and over again. So it's very important. It generates free phosphate, right? So it's important in that process. So we spend a lot of time, and don't worry, I'm not going to go all the way through this. We spend a lot of time trying to map out pathways associated with caveolin-1. What are caveolin-1 interactors? What are things we might look at to maybe how caveolin-1 forms these vesicles? This is work currently being led by Sophie Ashbrook, who came from Mississippi State, who Dr. Chen did as well, and joined us last year in the lab, looking at some of these mechanisms. But what we've really keyed in on is the fact that one of the closest interactors with caveolin-1 is a protein called EGFR. If you work in the cancer field, you might know the EGFR as well. So EGFR is commonly targeted in different cancers. EGFR tyrosine kinase inhibitors are commonly used. Okay? And so because there's approved therapeutics, we became very interested in, can an EGFR inhibitor perhaps how these vesicles form? And so then for this context, we went away from atherosclerosis. We went to a chronic kidney disease environment. So we take mice, we feed them a high adenine diet for 6 weeks. Okay, so that's this here, all right? And that high adenine diet destroys their renal function. You can see all these vacuoles kind of in their kidneys here. This is a section of a kidney. And then we give them a high phosphate diet, which induces vascular calcification. This is very common. Again, you saw that subset of patients earlier in CKD, that calcification, especially when these patients start going on dialysis, if they form that calcification, it greatly influences their mortality, right? And then in some of these mice, we supplemented that high phosphate with this EGFR tyrosine kinase inhibitor, right? I want to note here the EGFR tyrosine kinase inhibitor did not change their renal function. We have a lot more measures of this than just the simple histology, but it didn't seem to mess with their renal function. So it's not improving their renal function, but what it does is almost completely prevent the vascular calcification, right? When we add the high phosphate. We're excited about this because unlike the atherosclerosis group, everybody in this room probably has at least some kinds of atherosclerosis right now. Some of us will go on and develop something that becomes problematic. Some of us won't, right? But we almost all have some fatty streaks probably in our arteries. But CKD, you kind of know as a person progresses through this disease, you might be able to figure out when should I start treating this individual, right? You kind of have a more controlled patient population. And they're very much in need of these therapies. And so again, you're seeing that this inhibitor reduces calcification in these mice. This is a cross section here. This is Elozin Reston. You can see how had the calcification burden in the medial layer of these arteries gone with this EGFR inhibitor. So we want to try to understand, why is this happening, right? That's great, but how is it happening? Why is it happening? So we return to our cell culture and show, just in cell culture, it's even more dramatic. We completely inhibit the formation of calcification by smooth muscle cells that are in culture. But what became interesting for us was that we're not inhibiting those smooth muscle cells from becoming osteoblasts-like. They still acquire all these markers associated with osteoblasts. So that was really interesting to us. In fact, if you look, some of these markers even increase, or at least have a trending increase by our EGFR inhibition, but they're not calcifying. They're not leading to that mineral deposition. We also see this in the animal. So if we take a cross section of those arteries, things like caveolin 1, which I told you is important as vesicle formation, it goes up in the CKD case compared to the control case, not inhibited by the EGFR inhibitor. Alkaline phosphatase goes up in the CKD case, not inhibited by the alkaline phosphatase inhibitor. But again, the calcification is decreased. But what is inhibited, I told you these vesicles seem to be very important for nucleating that mineral. And I told you that and you believe me because I guess I have the authority today. But that's actually been fairly controversial in the field, whether or not these are involved in this. And what we show here is that the inhibitor decreases the loading of those components into the vesicles. Right? So this was kind of big, not just in terms of thinking about a therapeutic, but also understanding that these vesicles indeed are important in that calcification process. So we're not inhibiting their formation in the cell, but we're inhibiting whether or not they're released in these vesicles. And so we've started looking at caveolin 1, which I told you is important in making these vesicles as it moves through the cell. And the caveolin 1 is getting trapped inside the cells, and it's kind of between actin. This is caveolin 1's in red here. We have actin in green. It's one of these very interesting rosettes that we really don't quite understand yet. We don't even know what the implications are of this yet. Is that a good thing or a bad thing? We don't know. We know in terms of calcification, it seems vascular calcification seems to be good. But it seems to be changing how that caveolin 1 moves through the cell, which is inhibiting calcification. So it's giving some pretty new fundamental knowledge on vascular calcification. But in the cardiovascular calcification world, whenever we make a treatment, the question we're always asked is, well, what about the bone? Right? Because if you're decreasing calcification on the vascular side, you don't want to decrease a person's bone mineralization, right? So we checked that in the mice, and in the mice, their bone density, so the chronic kidney disease actually decreased their bone mineral density, and the EGFR inhibitor increased it. Not quite back to normal levels, but at least over what the disease case was. And this is interesting because there's something known as the calcification paradox. I want to come back to this. But the calcification paradox has been shown mostly in women who have osteoporosis, because women are more likely to osteoporosis than men. The more severe your osteoporosis, the more likely you are to have vascular calcification. There seems to be an imbalance between these two tissues. And so we're reversing that a bit. And so this is what this shows. So this is, again, I think this study was done only in women, but the percentiles of aortic calcification is shown here, okay? And the bone mineral density is shown here. So you can see as your aortic calcification increases, your bone mineral density tends to decrease. Okay? So for a long time, this has been known for probably about 25 years or so now, this clinical observation. And most people say, well, the reason this happens is that your bone mineral starts breaking down. And that calcium and phosphate then leaches into your blood and ends up on the vascular. That's been one hypothesis. And there may be some truth to that. There's been some people who suggest that, well, people who have these pro-inflammatory environments just cause this pathological remodeling in both tissues, right? So they're not really connected in terms of the mineral. It's just that both of them are remodeling in a bad way, right? And there may be some truth to that as well. So there's been a lot of folks who've looked at this, but nothing conclusive. But one thing that I felt we came across a study as we started focusing on caveolin 1 that I thought was really interesting, and this is in caveolin 1 knockout mice. Okay? So this group made mice that don't have caveolin 1 anymore. And what they showed was that those mice have increased bone mineral density. For us, knockout mice means decreased vascular mineral. There's no reason to think it would go the other way around just by knocking out a protein, right? You don't have mineral leaching from your normal vasculature into your blood that goes to your bone, right? So that's just maybe there's something more fundamental here, right? And so same thing here. They knock down caveolin 1 in culture, and their bone cells, the osteoblasts, increase alkaline phosphatase. Whereas again, it did the opposite. So we've been starting to focus on this a bit more. This is a PhD student who just started this past year, Catherine Kaiser, who's looking at this calcification paradox and where there's like some fundamental cellular differences in how these two cells mineralize that might explain that paradox. So this is repeating a bit of those studies, the ones we've done before in that group. This is looking at endpoint calcification. So you can see when we knock down caveolin 1, we get no calcification in our vascular muscle cells. Here, it didn't seem to change it in the bone, so it certainly didn't deplete the calcification in the bone cells. We have some indication now that we're working to finish this up, but it's actually happening faster in the bone, right? So we reached that endpoint faster. So this is at the endpoint, right? So we started saying that's interesting. This one protein seems to cause these cells in culture, they're not talking to each other, to go opposite ways. So I said, well, what if we start messing with these caveolar domains, those little caves where the caveolin 1 sits? How does that change things? So the first thing we did was just took a sledgehammer approach. Okay? These caveolar domains are rich in cholesterol. So we used a compound called methyl beta cyclodextrin. It just soaks up all cholesterol in cells. So it destroys these domains, okay, and liberates caveolin 1. When we did that, our vascular muscle cells calcify more. Our bone cells don't calcify anymore. The vesicles show the same response. So I've mentioned these vesicles are released. The vascular muscle cell vesicles have calcification potential. The bone cells vesicles have decreased calcification potential. So then we said, well, is there a way to stabilize these caveolae? So we just destabilize them. Can we stabilize them somehow? So caveolae link to the actin cytoskeleton through this protein known as filamin A. And filamin A is normally cleaved by a protein known as calpain. Okay, so calpain cleaves filamin A. So what we did was we used a calpain inhibitor to try to stabilize those domains. Right? This is still very early work. We're still trying to figure out the specificity of something. But when we do that, this muscle cells completely inhibit this muscle cell calcification. And our reviewers made us do a non-parametric test here because we only had n equals 3, so it's not quite significant, but a trending increase in calcification on the osteoblast side, right? So again, exact opposite, just by messing with these domains. So then we said, that's really cool. So I have a good collaborator up at the University of Indiana, Bo Kamchuk, who does a lot of GWAS studies, population-based studies. He looks at single nucleotide polymorphism, places in your DNA where you have these single nucleotide changes, right? How that associates with whatever you want to look at, right? Any question you ask him, he'll go and look and see if there are any SNPs related to this. So we said, well, let's look at caveolin 1. So the caveolin 1 gene, the open reading frame is in red here, okay? And we looked at 1 megabases around the caveolin 1 gene. Right, and identified all known SNPs in those regions, right? And went to 2 different studies, the MESA study, which has a lot of coronary artery calcification data, UK Biobank study, which has bone mineral density data, and looked at these same SNPs across these different populations, right? To see if they have differential effects on coronary artery calcification, cardiovascular calcification, and bone calcification, okay? And we found some really interesting results. So within the CAV1 locus itself, 11 of the 14 SNPs we identified have an inverse coronary artery calcification bone mineral density relationship. Interestingly enough, in this one, all of them show decreased prevalence of coronary artery calcification, increased relative bone mineral density to the mean population. If we look 1 megabases outside that SNP, this is just a random few, just show that we also can have it go the other way around. So we have some that have increased coronary artery calcification and decreased bone mineral density, and so on and so forth, right? And in that case, we found 110 SNPs associated with the decreased coronary artery, increased bone mineral, and 40 SNPs with coronary artery, increased coronary artery, and decreased bone mineral density. So most of the SNPs we found in this region led to an opposite response in mineralization of these 2 tissues, right? So again, going along with both our in vivo work in the mice, the cell culture work, and now human population-related work. So we like to think about mechanics a lot, and I have a bit of a bias here, but I'm wondering, this is something that I'm going to kind of just give a hypothesis here, which we're starting to study now. Is this mechanically driven? Because if you think about how mineral forms in these tissues and when it tends to form, about the bone, so we know that bone mineral forms more when you compressively load it. Right? Cardiovascular calcification, one risk factor is hypertension. Right? So you're talking about increased compressive loading versus increased tensile loading. right? And caveolae and caveolin 1 are mechanotransductive. So one of their purposes is to buffer the membrane from sudden changes in mechanics. And we wonder if there's a set point in each of these two cells in terms of compressive versus tensile loading, and we're modulating that set point when we change caveolae, and it's causing them to go opposite directions, right? So that's something we're starting to study. We've started so far on just the vascular muscle cell side. This is work that was led by Mohammed Shever, who recently actually defended his PhD this past fall, and now working in industry, where he was just taking vascular muscle cells and putting them under a cyclic mechanical loading using a flex cell device, collecting their vesicles, and looking at their calcification potential. What he found was that when he stretches cells, so just in a normal culture medium, just the application of stretch increases the release of caveolin 1 into these vesicles. If he provides a pro-osteogenic environment, that's further increased. And interestingly, the alkaline phosphatase, that protein so heavily involved in calcification, just the stretch alone led to an increased release of this active alkaline phosphatase in these vesicles, exacerbated by this osteogenic environment, right? So showing that, again, that the stretch can perhaps motivate the release of these vesicles and cause calcification. We're not exactly sure, again, the implications of that yet and the relation to the bone, but that's something that we're focusing on. All right, now, so I've told you a lot about the vascular side, right? And the mechanisms driving what we look at, right? We're trying to figure out how can we prevent vascular calcification? How can we treat vascular calcification once it's there? But a big thing that we always like to think about is how do we find these patients in need of treatment in the first place? And some of this is motivated. We've been lucky enough to be part of a big NSF Engineering Research Center at FIU, which is focused on developing technologies for underserved populations in cardiovascular disease and diabetes. And the problem here is that most people don't go to the doctor, and the first time they know they have a coronary artery disease, right? That's 42% of all people who have coronary vascular disease. And for a lot of people, they lose their life, right? And for many of the rest of them, they have permanent cardiac damage. Right? This is especially problematic in low resource settings, where going to the doctor might not be the easiest thing in the world just because maybe you're short of breath. And that tends to exacerbate the problem. So our goal is to develop some point of care technologies to help identify this. And we're really focusing on relevant biomarkers that can tell us something about your stage of coronary artery remodeling. I'm not going to go into details about these different proteins that we've really started looking at. But what we're wondering is, can we say, follow a person like at a point of care clinic, at a community clinic, say, at this point, you need to go and see a cardiologist. Right? And we can help triage patients early on. This is never going to replace standard diagnostic techniques, but it's something to help us triage patients. And we have a panel of biomarkers that are helping us do that. Lynn Tong, who's working very closely with our collaborators at UCLA, so Dino DiCarlo is leading a lot of this effort, is making some microfluidic devices where the idea is that we take human, probably blood, and we're still working on exactly what we'll use, whether it's serum or whole blood, or how we'll process this. And run through this device and basically just use a simple ELISA technique to then look at the relative expression of these different proteins. And so that's something that we're actively interested in. Similarly, a lot of these patients, especially with the chronic kidney disease and diabetes, have peripheral vascular disease. And you might know this in terms of things like the progression of diabetic foot ulcers, amputations often in these patients. And there's not really a great way to monitor blood flow in those patients as well in the peripheries. So this is work with Dr. Anu Gadarvardi in our department, who does a lot of optical imaging. She's developed these cell phone-based modalities to look at tissue oxygenation. And so we're taking advantage of that and saying, how does tissue oxygenation change as a result of underlying vascular remodeling? So Asma, who just joined us this past fall, is working on this project, looking at how when we take a limb, in this case a mouse tail, so it's kind of a limb, and we occlude it and then release it, how does that response change whether or not we have calcification present, or whether or not we have other types of remodeling present? And we're seeing some really interesting responses where the change isn't as drastic in the mice. I almost said the folks. I think of them like little folks. In the mice with the vascular disease, because they have this really hard incompressible, well, incompressible is the wrong word, but their capacitance of the vasculature is decreased, right? So when we squeeze, we don't shut it off as easily, right? The blood flow. And so we don't see this same response in a normal individual. So we're trying to move forward and see if we're actually going to be starting some clinical trials soon with our friends at Baptist Health in South Florida to assess some of this in real patients as well. All right, that's the vascular side. So I told you that targeting relaxin might reverse existing calcification. EGFR inhibition might change caveolin-1 trafficking, might be a good therapeutic for people with chronic kidney disease. That same pathway might be very important in bone vascular homeostasis, and that we might be able to use serum biomarkers and optical imaging to help diagnose and find people who are in need of treatment. So I'm going to switch gears and go to the valve side now. So as I mentioned, the aortic valve, all your heart valves are very dynamic tissues. 3 billion times in a lifetime is what I told you, it opens and closes. And it relies on a very precise extracellular matrix structure to be able to do that. Where mainly the main proteins that are required for that kind of load bearing are elastin, the elastic fibers that are oriented radially. So this is the aortic valve. We're looking at it from the aorta. These are the elastic fibers in this direction. That allows those leaflets to stretch when it closes and touch each other, so the blood can't go back into the heart. The collagen fibers are oriented in what we call the circumferential direction, this way, which gives that valve leaflets and this tensile strength needed to keep falling back into the heart and prolapsing, causing blood to return to the heart. Right? And this is what it looks like by histology here. And during disease, this all gets messed up. So this is just showing what it really looks like, not just a cartoon. So this is from a mouse leaflet. You can see these elastic fibers predominantly in this direction. Kind of depending on where you are in the tissue, you can see collagen fibers mostly orthogonal to those elastic fibers. You can see this nice crimping of the collagen fibers. When it's unloaded, those collagen fibers are kind of crimped. But again, when it's loaded, those collagen fibers are like ropes. They become taut and don't allow the leaflet to move anymore. Okay. So because I'm an engineer, I feel like I have to show at least a stress strain curve somewhere. And that leads to this kind of really nice anisotropic response to the tissue, right? So Dr. Chen did a lot of this in his undergrad work, taking these tissues and looking at these biaxial these biaxial differences across the different directions. But again, that radial direction can stretch a lot. This direction can't. It's the main moral of this story, right? So calcific valve disease, and so similarly to the vasculature, this valve can become calcified and become very fibrotic. And you can imagine that structure is so important to that function. This is bad news, right? The heart then has to work harder to open it, and again, it can't close as effectively. And a lot of this seems to be from birth. So we know a lot of the risk factors for vascular remodeling. Okay? They don't hold as well for valve remodeling. So we know that there's a genetic predisposition in a lot of folks that leads to valve disease later on in life. So this is a viewpoint that we got to write last year in the New England Journal of Medicine. That was, I guess, 2 years ago now. It was a lot of fun. But looking at this kind of genetic predisposition and trying to bring awareness to it, because I think a lot of people, it's underappreciated. There was a study a few years ago that came out that said that if your sibling has valve disease, your hazard ratio of getting valve disease is something like 34. Whereas if your spouse has it, it's like 1.2. Right? That just underscores that genetic predisposition that must exist for that disease rather than environmental causes. Right? So we're trying to understand, with that development in mind, understand the development of the valve is very important. How does that valve develop in the first place? And then how does that then lead to disease later on? So this is just one of my PhD students showing off. He made these slides for me, and he's going to show how he can open up these mouse valve leaflets. They're tiny. So a mouse heart is about the size of maybe even smaller than a kidney bean, right? So what you're looking at now is just the top half of the heart. So he's already cut off the ventricles here. And the valve leaflets, it may be hard to tell. You can kind of see the commissures there. So they'll be inside the closed tube here, inside the aorta. And so we make an incision and we open them up. And when we do that, we can expose all 3 valve leaflets. So we have 3 here, the left, the right, and the non-coronary cusp. So left coronary cusp, right coronary cusp, non-coronary cusp. They're named based upon their association with coronary arteries. So the arteries that feed your heart originate from the aorta, right behind the valve, right? So in the left, it's hard to tell, but you have a coronary cusp here. So that behind that valve, there's a little hole there that blood will then flow back into the heart, right? In the right, you have a hole here. That's the right coronary cusp, and the non doesn't have a coronary artery associated with it. So 3 different leaflets. Okay. So we take those leaflets and we start looking at their structure. And this is work that's being led by Peroni, who's a PhD student in biology, and it's a collaboration between me and Lydia Koss, who was a professor in biology, recently moved to take the assistant provost position at UC Riverside. But she was on my my interview committee when I was interviewing for my faculty position at FIU. And she said, you know, in mouse aortic valve leaflets, there's melanocytes. So she's a melanoma person. And I said, I know, I hate them, right? Because when I'm trying to do histology, the pigment gets in the way. All of us in the valve field hate them. And she said, well, you know, melanocytes are a neural crest cell. They come from the neural crest. She said, no one ever looks at them. What are they doing, right? And I was like, I mean, my back of my mind, like, who cares? Because we don't see them in human. We don't see pigment in human leaflets, right? I was like, well, you want to send me some hearts. I was still working in Boston at the time. We can do some imaging and look at their structure. Right? And so she had wild-type mice here, hyperpigmented mice, and hypopigmented mice. This is a KIT deficient mouse, but we have other models we've looked at as well. So you can appreciate the difference in pigmentation across these leaflets. There's one leaflet here that you're looking at. So this is how that structure of the normal mouse looks. You see this radially aligned elastic fibers. This is all done by 2-photon microscopy, the circumferentially aligned collagen fibers. Pigmented mice, we saw an expansion of their elastic fibers, right? It's all kind of messed up though. In the hypopigmented mice, we could find no elastic fibers in their valves, which was wild to us. So no one knows how elastic fibers are made in the aortic valve, to the point where there's a paper by Catherine Yutze, who's a big person in the developmental field a few years ago, that said that she did a single-cell RNA-seq paper, and said they were unable to identify the single population responsible for elastic fiber formation. So these cells might be extremely important. Okay? So we're doing a lot of work now into the mechanisms that we still don't know what's going on, is the short answer there. But these cells we think are extremely important in just formation of that valve from the very beginning. Okay? So if you were really perceptive, so this is Daniel who put together those first slides. He's also a senior PhD student in the lab. If you were perceptive, when I showed you these three leaflets here, you might have noticed that only 2 of them were pigmented and 1 of them wasn't. So he's, at this point, probably isolated 500 of these mouse aortic valve leaflets. And he's really diligent in keeping up with, this is always keep up with your notebooks, very, very nice notes of like what you're doing, what you're observing. He was keeping up with how often he saw only 2 pigmented and how often he saw 3 pigmented. And the ratio, so 70% of the time we see only 2 pigmented, and 30% of the time we see all 3 pigmented, almost exactly. And when he came to my office one day and told me that, I almost fell on the floor, because that's the exact penetrance by which enos deficient mice, which is a common mouse model in the cardiovascular field, those mice develop bicuspid aortic valves. So instead of being born with 3 valve leaflets, they're born with 2. This is the most common congenital valve defect in humans, or heart defect in humans, 1 to 2% of the population. Okay? And people with bicuspid valve malformations, about 50% of them go on to have valve disease. So he told me that, so I was like, well, let's go look, let's buy some enos mice and see if when they have 3 leaflets, do they only have 2 of them pigmented? And they did. And sure enough, when they only have 2 leaflets, both of them are pigmented. So we think it's some asymmetric distribution of these cells that, again, important in elastic fiber formation, also apparently important in how these leaflets separate from each other during development. So now he's crossing these enos mice into the mice with a different level of pigmentation to see if it changes whether or not they form those 2 or 3 different leaflets. Okay. Again, some of this, Danny likes to show off sometimes, so some of this is just him to show off. So recently, he's also been looking at cardiac function. He's basically become, him and Valentino, I'll talk about next, have become mouse cardiologists. So we just got a new Vivo F2 ultrasound system. So we're also able to now look at cardiac performance in these mice and understand how, when we change that structure, how we alter how blood flows in the mouse. So you're looking at blood coming out of the mouse heart into the aorta, filling the carotids, and we can nicely then also not just look at structure, but how that structure associates with function, and then how that function changes as we progress toward disease. And so that's something we're really excited about, again, connecting development all the way to disease in the valve. My last couple of slides here. So just like in the vasculature, maybe even worse, valve disease goes extremely underreported. So valve disease progresses over the course of probably 2 decades at least. And the symptoms of it are dyspnea, so, shortness of breath, fatigue, things that if you're in your 50s, 60s, 70s, I mean, there's a lot of days I feel very tired right now. You mistake it's just signs of aging. And people who end up with a valve replacement, some of them say it's kind of like, those of you who wear glasses and wear contacts, the first time you put on those glasses or contacts, you didn't realize you couldn't see before. They say it's a lot like that. They didn't realize they couldn't breathe before until they had this valve replacement done. And again, that's also if you have to work 2 jobs, if you don't have adequate access to health care, going to the doctor because you're short of breath is probably not something you're going to do until it becomes very severe. And when it becomes very severe, that means your heart muscle is compromised as well, right? So there's a huge need to find people early on with this disease. Okay. And so this is where Valentina comes in. So she's a PhD student in the lab, but this idea originated somewhat with my wife, who is a classically trained opera singer. And so when we were at Vanderbilt studying valve disease there, she was studying at Belmont University, studying music, but she was working some of the Ear, Nose, and Throat Institute at Vanderbilt. And I was always interested in how the vocal folds look a lot like your aortic valve. And we were talking about how you get these pathologies on your vocal folds start thickening, or you get nodules forming, how that changes the frequency of how your voice sounds, right? So the idea was, if that's the case, well, then if you get a small structural change in your aortic valve, your heart sound should change, because that loved up, that S1, S2 sound that the doctor is listening to when he or she is listening to your heart, that comes from your valves, right? Even if it's not perceptible, now that we can do digital recording of heart sounds and use things like machine learning, we should be able to find small changes in that pattern that might tell us something's going on there. The only other way to diagnose it now is with echocardiography or some large-scale imaging that's, again, not going to be deployed on a person who's not reporting symptoms in a general practitioner's office. This is a way. You go to your doctor, one of the first things he or she does is put the stethoscope. Right? If we could adapt that, we might be able to find people who need that further workup, even if they don't know they need it. Right? So again, Valentina has become somewhat of a mouse cardiologist. So turns out there's no mouse stethoscopes out there, so we were able to take a human stethoscope and adapt it. We put these little bumpers on there and put it on a little mouse chest and record their heart sounds. And so we use the same mouse model we used before, these chronic kidney disease mice. Again, we feed them a high adenine diet, feed them a high phosphate diet. Valves calcify, just like their vasculature does. And we recorded their heart sounds. So the mouse heart beats at 400 to 600 beats per minute, so a lot faster than ours. And record their sounds, and then using an ensemble-based machine learning algorithm we've developed in collaboration with Dr. Zach Danziger, who was in our department, just recently moved to Emory, we showed that when a mouse is a control mouse, we're able to classify it as healthy 97% of the time. When it was just given the adenine diet, so no calcification, we're able to classify those well about 90% of the time, and about 85% of the time for the calcification. So there's 2 interesting things here, right? One, we can triage patients who don't need further workup. So we can easily tell you do have a problem versus you don't. But even with these 2 subsets of problems, we might be able to tell differences there. And we're now trying to figure out what is it in the signal that this is picking up. There's actually some heart failure issues there going on. There's common CKD that we think we're grabbing there. So this indicates that we might be able to use those heart sounds, again, as a very low cost, non-invasive device to find people who need this further workup. Just going to close the loop here. This is my last data slide. So that EGFR inhibitor that seems to work so well in the vascular calcification, I just got these data last week. It's very noisy for now, but we're adding more mice to it. Might also work in the valve calcification as well. So we're trying to figure that out. We're adding more mice to this. That would be super exciting as well, because now you can then think about finding a person using our techniques early on that might need treatment, referring them to more clinical workup, and then we're actually having a way to treat them, because valve disease is currently untreatable. Okay, so in the aortic valve, to close everything up, there's these weird atypical melanocytes that might play an unappreciated role in valve structure. We have gone back now and shown that these cells exist in human leaflets as well. They don't make pigment for reasons we don't understand, but they have all the classical markers of melanocytes. So, interestingly to us, I was at a conference 2 weeks ago where one group that's studying bicuspid valves, I told you this melanocytes might be important in bicuspid valves, they were doing single cell RNA bicuspid versus tricluster valves. One of the major groups that was different between the two were they classified as melanocyte-like cells. So again, further indication that what we're seeing in the mouse might also be relevant in the human. Right? If we understand that BAV phenotype, we might find people with valve disease, also using our auscultations and allow for early diagnosis and perhaps treatment. So this is some of my group here. A lot of good collaborators. We've been fortunate to have support from the Florida Heart Research Foundation, NSF, NIH, AHA. You're welcome to follow us on social media. So I have a still called a Twitter account. That's run by me. If you want something more fun, the Instagram account is run by me. So I don't necessarily endorse everything they do there, but it's at least a lot funnier than what I put up on Twitter. So, but thank you for inviting me.
Questions for Dr.?
Sure.
It's a great talk, very fascinating topics. Thanks for sharing that. I was thinking about when you're talking about the bone structure, change in the bone structure compared to calcification in the arteries. And I was thinking about astronauts that go to space, on the space station for 6 months or something like that, and they actually experience sometimes quite a bit of bone loss. That when they come back to Earth, their normal gravity tends to kind of come back to normal. But if you have an issue with abnormal mechanical stress in the arteries, that's addressed and returns to normal, do you eventually see the same types of changes, or is it different than you would see maybe with like the bone density in the astronaut?
So are you asking if the astronauts have issues returning to normal if they have hypertension, or just
this process?
Sure.
Because obviously the astronauts are experiencing changes in the mechanical stress or load, I guess. On the
sure.
bone.
Yeah.
And in the case of someone has hypertension or something that's causing increased stress on mechanical stress on the artery, on the smooth muscle or arterial wall, if you change that, does that basically help with calcification, or is it basically staying that way? Because obviously with astronauts, their bone does
regenerate. Yeah. That's a good question. The reason I asked if you were asking about the astronauts themselves, because that's actually one of my, I want to write one of these grants to be, for research to be performed on the space station, the CASIS grants. So I've been thinking about that in terms of bone vascular paradox. But yeah, so you're saying if you unload, like able to manage hypertension, does that decrease calcification? I don't think anyone's done that study carefully, I don't think anyone's actually looked at that much, right?
That would be what you were saying about the possible mechanical stress being a key factor.
Yeah.
So that's why I was thinking about the astronauts. They go through that similar transition, and I wonder if that could be a therapeutic. I mean, obviously, reducing hypertension is already a therapeutic strategy, but I wonder, how much of a role that.
It's a good point, a good idea. I think a lot of times when people are undergoing hypertension therapy for hypertension, they're pretty late in life anyway. Well, they probably have very significant vascular. They might not have long enough to reverse that. I would be a pretty good control for that because I'm on losartan now, so I take a small amount of losartan because I have hypertension that runs in my family. And so I wish I would have gotten a calcium score done before I started taking it. Yeah, that would be interesting. At what point with that unloading could you also get resorption of that mineral?
Calcification, of course, can be involved in a number of health conditions outside of cardiovascular stuff. Do you see this being applicable to that calcification process also?
Maybe is the short answer. Yeah, we haven't looked well enough, but we've had some groups we've talked to recently about calciphylaxis and things like that. So like when you get calcification basically all over, it really shows up in your skin a lot. And so we're going to, we actually have a meeting in with a group to see if some of their models, maybe we'll try some of the treatments. And just, the first thing is to see the similarities and differences between those types of calcification and what we're seeing on the vascular side. But I can tell you at least, I suspect it's going to be the same, right? Because they're seeing all these kind of bone markers go up, and these are usually fibroblasty mesenchymal cells that are probably doing something similar to what some muscle cells are doing in terms of acquiring that phenotype. So I think that's going to be interesting. And there's kind of one other point to that too, in that a lot of people who study things on the bone side use mesenchymal stem cells to push them toward an osteoblast. We started looking at that just a little bit, and those cells tend to follow what we see in the smooth muscle cells. Whereas if we use primary osteoblasts, they don't, like I showed, right? So I don't want to like say something too controversial about the whole bone field, because there are people who use primary osteoblasts, but I do wonder if there's a major difference in these mesenchymal fibroblast-like cells that try to push toward an osteoblast versus a primary osteoblast that may have developed very early on during embryogenesis. So that was a long way of saying I think that we'll see something similar in the osteoblasts. Yeah.
So when you're talking about looking at these heart defects with these INOS, you said nice and everything, and kind of finding that correlation there, and your collaborator had alluded to the on-the-side being part of that kind of early developmental process. I was curious, in humans, there's a rather significant portion of children that are born with various degrees of heart defects. I mean, I had a hole in my heart when I was born, gives murmurs and all these other defects just going on.
Yeah.
I was curious if there's, to your knowledge, any studies or reason to really look into this kind of development or this state that many grow out of or becomes non-serious, if there's any link between calcification and
well, I think certainly these cells I think are playing some role in like septation of the heart, so we're the only group looking at these cells. I can tell you, for better or worse. It scares me sometimes. Like literally no one else. We're the only ones. But we have worked with Elaine Shelton a little bit. This is before the pandemic, and this reminds me to pick it back up with her. But she studies patent ductus arteriosus, right? This, the right side heart closure that happens when most babies like when they're born, right when they're born, and some don't, at least a blue baby syndrome, right? So she has a mouse model of that, and the reason she contacted us was that mouse model said it's a point mutation in a potassium channel. That mouse model, when she looked at his aortic valve leaflets, they were pigmented like crazy. So again, what that means, I don't know, right? Like, but to me, it means something about these cells, these mechanisms that we're studying that are very important in formation of the heart from the very beginning of septation. And then we're looking at how reemergence of the same pathways that might play a role in disease, but that's work that's still ongoing.
Follow that question, Josh. So these melanocytes, and I think it's interesting, work that you're showing on them, but is there some way for you guys to sort the cells from these valves and isolate these melanocytes and do some, rigorous experiments to kind of figure out what they do?
We're trying. So Catherine Yuxey was able to do single cell analyses on mouse valves, and she sees the population of melanocytes there. The problem with these tissues in the mouse, at least, are so small, right? And so culturing them will be a major problem, I think. So we're trying to see if we can, at different developmental stages, do what she did in terms of at least doing some single cell, omics of and what they are, what they're doing, how they progress through development. Even that's just so hard to get. Appreciable cell, she had to combine 10 mice and combine all heart valves together and so on and so forth to get one data point basically. That's something we're thinking about doing. For those of you who does iPSC work, one thing that we've thought about lately too is trying to see if we can kind of recreate. So the valves are a mess. So the cells that make up those valve leaflets come from the neural crest, they come from the endocardium, and they come from the second heart field, right? And there's ways to push iPSCs, different lineages, to these kind of endpoint cells that we're seeing there. Maybe it's maybe something like making an organoid based system that kind of recapitulates something about that valve cellular structure might be of interest. But
I'm curious because the fact that it's pigmented indicates that there's some specific pathway that maybe leads to pigmentation that there's.
Right.
separate them.
Yeah,
But I think that, yeah, I'm sure.
it's just so
much more
few cells.
challenging
Is
than-
and the pigment seems to be extracellular, right? So the melanocytes. So classically, in your skin, melanocytes release pigment, and they're taken up by keratinocytes. We don't have keratinocytes in the valve, and that pigment just seems to be floating around the valve matrix. So yeah, but what we do have though from Lydia's mice, she has a GFP reporter driven by tyrosinase, which is the main protein that makes pigment. That's tyrosinase gene. So we can get fluorescently labeled melanocytes. That's what we're trying to use now to see if we can separate them. But it's been difficult.
Changes that molecular velocity and all this stuff are related to HPA changes in What are the difference with the HPA conditions?
Yeah, no, I definitely think so. So most of the things that we study are age-related diseases, and I think that age-related changes are playing a role. What those are is unclear, but there's a lot of people who are looking at cell senescence now, and its role. So I think that's really interesting that we haven't really explored. I didn't even talk. So there's surprisingly enough, there's a few things I didn't even talk today's talk, but there's also a population of neurons in the aortic valve that are also neural crest derived. There's only one other group that's looked at those. We know that those decrease in abundance with age, and they seem to be very important in controlling valve tone. So that might be affecting the mechanics, it might affect the remodeling. So I think there's some age-related processes that are certainly affecting it. We'll understand those well.
There it is, pizza up front, where they want to do.
Yeah. Back up.
Let's put something up.
You want this one?
Yeah, whichever one you think for you. Or, you want to stand next to it? Just try it. That one.
It's a little crazy. It's, I was like,
We're just handling like 3 or 4 and something like that.
But yeah, there are like games.
Okay, I can go there.
All these things on hyperdrive.
Okay, perfect. Okay, I'll just meet you there then, because I'm making my back.
She doesn't have.
No, we don't have one still.
A lot of, and it seems to be a mechanical thing where holding things together like where they're supposed to be.
Hey, nice to meet you as well.
Were you together?
It's been having. It's been a while.